comprehensive, and you probably see a couple of the figures that we cited in there, citing in this PowerPoint. So
awesome, cool, yeah, take it away, dude.
All right, thanks. Let me just share my screen here, and we'll get started.
Nice.
All right. So yeah, thanks, Stephen, for all that those kind words. Likewise, Stephen's an awesome guy, and is smarter than I'll ever be. But you guys are lucky to have him as a professor. All right, so today we're going to talk about SARMs, right? So selective androgen receptor modulators for your drug education class. Time is of the essence, so we're going to kind of skim through a lot of it, go into a little bit of depth, but pull back a little bit so we have time to talk about the data at the end, which we are currently drafting up for publication. So let's get started here. So today we're going to talk about some basic androgen receptor signaling, what SARMs are, why they were developed, how they kind of infiltrated the fitness industry, whether that be recreational or competitive athletes, how they work, and what happens when someone takes them. So again, we can get into the intricacies of AR signaling, but I feel like we need to understand the basic principles behind it in order for us to better understand the SARMs. So and I'm going to be referring to the androgen receptor as AR throughout this presentation, so don't get confused by that. So for basic androgen receptor signaling here, we're going to have our circulating androgens traveling through circulation. We can go over at the end how we actually make these androgens through our HPG axis, but for the sake of this for now, we're just going to start here. So we have our circulating androgens, and these are lipophilic, right? These are steroidal in base. These are the basic structure is a cholesterol backbone. So these are lipophilic and can diffuse across that cell membrane, which we can see here, where it's going to interact with that indwelling androgen receptor. So currently that androgen receptor is in an inactive complex, an inactive conformation, and bound by a bunch of proteins called heat shock proteins. So once the androgen infiltrates that intracellular space and interacts with the androgen receptor, we're going to be getting some dissociation of those heat shock proteins, as you guys can see here, and then forming a active androgen receptor complex, which becomes an active DNA binding complex. So the fancy term for this is that receptor becomes transactivated and dimerizes, and then we can get what's called nuclear translocation. So that's going to be this activated DNA binding complex translocating through that nuclear membrane and into the nucleus where we have our DNA. So once it's in the nucleus, there is a specific sequence on our target gene called the androgen response element. And basically this is what triggers the upregulation and androgen regulated gene transcription. So when I say gene transcription, I'm talking about turning on the activity of these genes, which in hope will eventually get translated into proteins. And these proteins will become integrated into, in our case, the skeletal muscle, and then become biologically active. There's a number of AR regulated genes, not super important for the sake of this lecture, but a number of them have to do with increases in muscle hypertrophy and in increases in contractile proteins that were responsible for increases in contractile strength of the tissue, so i.e. strength, right? So now that we have a basic understanding of how AR signaling is taking place, we can introduce what SARMs are. So SARMs, like we said before, are selective androgen receptor modulators, right? So all of those words in that are pretty key to understanding exactly their actions within the body as a whole. So we'll start by, again, these are a class of anabolic compounds that can bind and alter the androgen receptor activity. So again, a key word here being alter. So because they are modulators and not purely activators, it depends on the tissue within the body on whether they can activate and upregulate this system, or whether that they can bind to and inhibit or block this upregulation in AR signaling. So that is a key characteristic of SARMs, is that they are tissue selective. So I'm going to hammer home this point because it kind of adds to and sets up whether it's how it's used in the clinical sense, which we'll go over, and how it's used in the fitness and athletic world. So because they're tissue selective, again, which I'm going to go over more, we're going to have ideally a preferential increase in anabolic properties of that tissue, but a decrease in the androgenic properties. So when I say androgens or androgenic properties, usually it's associated with the negative effects of androgens on a tissue. So when we think about, a great example for this is like anabolic androgenic steroids, right? So we have these strong steroid compounds, like let's say trenbolone or something like that, something that's very considered androgenic. We're going to be increasing a lot of these side effects and cross interactions with all these different tissues. So we see, alopecia or balding, we see gynecomastia, we see testicular atrophy in men, fluid retention, and a number of other negative side effects that are a result of excess androgenic activity. And of course, we can see this also, not just in males, of course, but in females too, when abuse of anabolic androgenic steroids are observed in males and females. We both, in both scenarios, we see increases in this androgenic side effects. So the idea behind these, again, increasing the anabolic properties while minimizing or limiting the androgenic properties of these androgen compounds. Something else I want to mention here, that these are orally bioavailable. So instead of injections that are commonly associated or commonly done with anabolic steroids, these can be orally administered and delivered to the target tissue, again, through the mouth. So on the side here, I just want to mention too that we have a number of different types of SARMs. Okay, so we have ones that are propanoic acid based, quinoline based, and a number of other bases here that are all lumped under the same name of a SARM. So it's important to know that when we talk about SARMs, there are a number of different compounds, and they do have differential and specific effects that are unique to them. Okay. We also have steroidal-based SARMs, which you could imagine would be most identical to a steroid-based molecule, like testosterone. So hopefully you guys have a little bit of that endocrinology background here. I'm not sure if you guys can answer me here, but out of these compounds over here, can anybody recognize the steroid-based SARM here?
Andrew said the bottom one.
That's correct. So that's YK11. So that one is a steroidal backbone, or a cholesterol backbone. We can see here by these four rings here, versus something like ostarine, which has been studied, probably the most studied SARM, both in cell culture animal models and in human data in low doses. That's a propanoic acid base when you see this propionamide here. So they're going to have differential effects. So it's important to note this. And we can see potentially how others can affect this spectrum of anabolic to androgenic effects. Okay. All right. So again, I want to hammer this point home because it's really important to how they work and to the advantage of these compounds. So they're, again, tissue selective. So I just talked about that anabolic versus androgenic effects of these compounds. So I'll start by talking about how they can do this. So to be honest with you, with all of these compounds, with the fact that they are all different and they are still somewhat in the early stages for development in terms of their clinical application, they'll never really get approved for recreational application, right? But for clinical application, so how are these compounds tissue selective? So most of these SARMs, if not all of them, are preferentially active in skeletal muscle and bone with limited or very small amount of actions on tissues like prostate. All right, so this is prostate down here. So how can this be? Again, we don't necessarily understand the intricacies of these mechanisms, but from the data that we have thus far, either A, it's going to impact 5-alpha reductase activity, or it's due to the fact that it's how that these compounds bind to the AR in their specific tissues. Okay. Does anyone know what 5-alpha reductase does?
Andrew said it creates DHT.
Exactly. Awesome. All right, you got some good students there. So perfect. So yeah, so 5-alpha reductase is the enzyme responsible for reducing testosterone to DHT. So DHT is similar to that YK11 molecule that we talked about a second ago, in that it's very potent, and it's about, again, DHT compared to testosterone is about 5 times more biologically active than testosterone. So it's important to note that even within the normal physiology of us, that we have degrees of activation of the androgen receptor, and that all androgens do not necessarily interact with the androgen receptor in a similar manner. So the same can be considered with these compounds. All right, so we'll talk about again later kind of how they bind to the AR and how this influences the conformational change of the AR, and then influences those downstream effects, meaning the transcription and then translation. So again, these tissues are selective, so they can act as agonists or antagonists. So in skeletal muscle and bone, it's going to be activating this receptor, and then prostate, to a certain degree, it's going to be blocking or antagonizing this. But I want to hammer home that this is a spectrum, and it's not a check mark, or it's not a light switch of on or off. Again, how these compounds interact with the receptor is going to be a degree of change throughout. So again, in bone and muscle, we're going to get that activation. In prostate, we wouldn't see much. So why were these SARMs made, right? So the idea starts from selective estrogen receptor modulators. A lot of the technology that was used to develop these was adopted into development of these SARMs. Does anybody know any selective estrogen receptor modulators by chance?
You said Clomid. I mean, potentially,
Yep.
if here has family members that maybe had breast cancer or something of that nature, you might have heard of Clomid or clomiphene. You might not know those medications either. Other ones are going to be things like Nolvadex. They're going to be other selective estrogen receptor modulators.
Exactly. So these SERMs, like tamoxifen was one of the ones, is the compound name for one of them that Stephen was mentioning. So tamoxifen is a selective estrogen receptor modulator. So in breast tissue, it can block the actions of estrogen on that receptor. But in tissues like in females, like in the uterus or in bone, it can actually activate estrogen receptors in those tissues. People also in the bodybuilding and fitness world have been known to use tamoxifen as a post-psychotherapy treatment to help kickstart that axis again. And that's because it blocks the estrogen receptor in the hypothalamus and pituitary up top regulation in terms of this axis, and basically tricks the body into saying, we don't have enough estrogen. How do we get estrogen? We can kickstart this axis to help increase testosterone, and then the conversion from testosterone to estrogen by aromatase would also take place. So we have this technology that we can kind of use to our advantage. Now, in the context of medicine, SARMs have been, again, manufactured and made for the treatment of prostate cancer, cancer cachexia, hypogonadism, a variety of muscle wasting disorders, oral contraceptive use, osteoporosis, right? And this is all due to its tissue selective nature, right? If we can, same thing with the concept of SERMs in breast cancer tissue with kind of blocking or inhibiting the actions of the estrogen receptor. In prostate cancer, one of the big, one of the primary mechanisms of cancer growth and tumor growth in prostate tissue is an androgen receptor regulated upregulation in tumor growth. So if we can block that receptor, in theory, we can decrease some of those negative impacts. We can go into some of these other ones here, like contraceptive use, like that kind of sounds, how does that fit into all of this? We could talk about that later. And actually some of our data kind of brings this up a little bit. And osteoporosis, again, we talked about it's active in bone, right? So increasing bone content through activating this axis could also help out a bit. All right, so like any compound, honestly, in the world that has been known to increase anabolic properties such as muscle hypertrophy, increases in muscular strength, and improving body composition, whether that be increasing muscle protein or whether that be decreasing in fat mass, recreational and competitive athletes are going to try to take advantage of this, right? So we see a very large increase over the past, I would say 10 years in SARM usage in recreational athletes and competitive athletes. Here in the University of South Carolina, I think up in Clemson, about 5 or 10 years ago, someone got caught using Ostarine on the football team. So they are starting to screen for a lot of these compounds because they are not FDA regulated. They are not approved for human consumption. These are strictly research chemicals at this point. So nonetheless, though, it's not going to stop competitive athletes from trying to take advantage of this, especially in the bodybuilding industry where testing doesn't really take place for these compounds. So again, it's important to note that the idea of this is if we can get all the benefits of steroids without all the negative side effects of androgen of those steroids, of those androgenic negative side effects, then this is a gold mine for physique athletes and recreational and competitive athletes, in addition to all of the clinical relevance that this has. All right, so how do they work? So this is a little bit busy, but again, some of you guys might recognize this from the paper that Stephen and our team published. So we talked about basic AR signaling before. So let's just go over that real quick here, right? So we have our cell membrane here, and that androgen, in this case, we'll talk about testosterone, is going to diffuse across that cell membrane, right? Is going to bind with the androgen receptor and fully activate that androgen receptor complex, which is right here, right? So we can talk about the different domains and the different aspects of the androgen receptor. So we have the ligand binding domain, we have the DNA binding domain, and we have the hinge region. All right, and within these regions, we have these, within these domains, we have these specific regions called the activation function region. So we have AF1 and AF2. So the AF2 is one of the big guys in terms of facilitating androgens and SARMs with interacting with the androgen receptor and causing that conformational change. So structure and conformational change in terms of these proteins, i.e. the androgen receptor, that's what's going to dictate function. Okay, so how these proteins are folded is going to help dictate how they function. That's going to regulate affinity. So think about affinity as a strong magnet in terms of pulling it to its biological function. So again, backing up here. So we have that testosterone interacting with this receptor, specifically in that AF2 region in the ligand binding domain. That's going to cause this NC interaction. So this N terminal and C terminal interaction that's going to fold this protein and make it a active complex. And that is now a DNA binding complex. So once that occurs, we have full activation here of this receptor in terms of the androgen binding to it like testosterone. And like we said, it's going to translocate to the nucleus here and bind to our target gene in a specific area in the promoter complex that's going to call the androgen response element that's going to upregulate transcription. Okay, so now we have increases in genes that are responsible for increases in muscle protein, both myofibrillar and sarcoplasmic, that's going to increase size and strength, and we can get eventual increases in anabolic and androgenic activity and eventual increases in protein expression. So hopefully that makes sense and you followed me through that because we talked about that earlier on, right? So now let's talk about the SARM here. So follow these red arrows. So same thing, these SARMs, they're lipophilic, right? We have, like we talked about, propanoic acid or propionamide. This is a fat-based molecule. We have steroidal-based SARMs like YK11. So again, these are lipophilic hormones or lipophilic compounds that can diffuse across that membrane and interact with our indwelling androgen receptor, right? So same thing, when the SARM interacts with that receptor, it's going to interact in that AF2 region, in that ligand binding domain, and that's going to cause this conformational change again, but it's going to be, depending on the compound, we're going to change the function or change the way it folds, right? So we might have what you would call incomplete, this NC interaction. So here versus here, if you guys can see my pointer. So there's still activation of the androgen receptor, and it still allows it to translocate into the nucleus and bind to that target gene, but it might not be to its full extent. So that's what we're seeing here, and that's going to increase transcriptional regulation of this tissue, but potentially reduced androgenic side effects or androgenic properties of that tissue, but in theory comparable anabolic properties of our natural physiological testosterone. So again, and this is what kind of lends to the individual actions of these tissues on the individual actions of these compounds on individual tissues, whether that be muscle and bone, or in prostate, we wouldn't be seeing this. Okay, so we got through a lot of the meat. Hopefully you guys have a basic understanding of how these SARMs work, the general principles of them, because that's what makes this data exciting. So I wanted to make sure you guys had a good understanding of that. So Stephen and I, during my thesis at Baylor, which we were investigating some AR related signaling changes in response to different training techniques, different resistance exercise load techniques, and we came across an individual who was taking a SARM. So you bet me and Stephen were like, yeah, you're going to stay in the study. We won't use your data in this analysis. But we wanted to find out what was going on here. So this 25-year-old resistance trained male, he was taking 10 milligrams of ligandrol. So ligandrol, or LGD4033, is a quinoline-based SARM, and he paired this with 15 milligrams of ibutamorin, or MK677. So this one is technically not a SARM. MK677 is a growth hormone and ghrelin secretagogue. So when I say secretagogue, I mean it enhances the release of these hormones. So if anyone knows what's the effects of ghrelin, what does ghrelin do? Does anybody know?
It makes you hungry. There you go.
Makes you hungry. Exactly, right? So ghrelin is linked to growth hormone secretion. And does anybody know why we think GH would be elevated when you're hungry? It's thought to be an anabolic hormone, right? Why would GH be upregulated in response to increases in ghrelin in circulation, or just plainly put, when you're hungry?
You said related to insulin in some way?
Insulin probably has a regulatory role in releasing some of these hormones, but the big reason is that GH is more lipolytic necessarily than it is anabolic. So it has to do with metabolism for sure, but it actually helps out with lipolysis and using fat for fuel. So we see in times of low substrate availability, right? You haven't eaten in a while, that your body's going to have to call upon some fat stores for some additional energy, and GH gets released. And we can go off on some of these compounds too, because they have a ton of clinical relevance and a ton of history of use and abuse in the athletic world. But anyway, so I say this because I want you guys to keep in consideration that some of this data that we see is not necessarily due to just the use of this Ligandrol, but it could be due to the additional use of this MK677. So he took these compounds daily for 5 weeks. No published research prior to this has looked at the effects of up to 10 milligrams daily of Ligandrol. So the previous data have only showed up to 1 milligram, and they saw some interesting decrements in some of the markers that we're looking at. So 10 milligrams is definitely going to be interesting to see which is more common in terms of what people take, the influences on skeletal muscle and some health markers. So
real quick.
Yeah, what's up?
For reference for everyone, do you know what the highest dose studied is of Ligandrol?
I believe it was so they had a point, it wasn't like a 0.0, a 0.1, a 0.3, and a 1 milligram dose of Ligandrol, I believe, in human data.
Absolutely, yeah. So this is 10 times more than the highest dose ever studied.
Exactly. Yep. And this is already an area that's not studied very much, number one, because again, this isn't approved for human consumption. And it really only got past phase 1 and phase 2 of clinical trials. Some of these other compounds, not this one. So again, this is an area that's not looked at much at all, but it needs to be. So before this guy came in, we had some data from before he took these compounds. So we had blood data. We did a comprehensive metabolic panel, along with like other health-related and androgen-related markers. And we also have DEXA data. So DEXA helps us analyze body composition. We can look at bone content and area and density. We can look at fat mass. We can look at lean body mass, and of course, total body mass. So for 5 weeks, he took these compounds, and then post-cycle, still on cycle, but post-cycle, if that makes sense. We also got additional blood data, and we took a muscle biopsy, from his vastus lateralis. All right, so from his quad, to look and see what's going on intramuscularly. Then he came off for 4 weeks. We told him to come back, and we got more blood and body composition data. All right, so this is the interesting stuff here, and I want you guys to kind of ask questions and try to interpret what we're finding here, because it's interesting, and there's definitely a lot of questions here that this data can't answer, but opens up a lot more new questions. All right, so what's happening in body composition? So we have an individual here. He went from about 97 kilos up to about 103, 104. So what's that for? So like 6-7 kilos, so 13, 14 pounds. Pretty substantial increase in 5 weeks. All right, so a hair over a month, this guy gained 14 pounds. Let's look and see how much lean mass that was. So muscle mass, about 78, 79 kilos up to about 83, 84. All right, so about 7 pounds of lean mass there accruing. So in theory, right, so these SARMs are activating this axis, and we're increasing muscle protein based on this data. However, in addition to that, we see an increase in fat mass. Now, it's important to pay attention to the scales here. This goes from about 15.8 up to about 17.2. So we'll say little shy of 2 kilos, maybe a little bit more than 2 kilos, right? So about 5 pounds of fat mass.
Hey, Tom,
And then,
you know that
what's up?
You got like 5 minutes. Well,
Okay.
we're at 9:20, but you know, if they want to ask a couple of questions, just letting you know.
Gotcha. All right, I'll run through this part then and get to the good stuff. So, and then bone density, not much changing here, we'll say, although a small decrease. All right, so this is some of the cool blood data that we have. So just to orient you guys here, we have this is pre-cycle here in the gray, immediately post-cycle in the black, and in the red, we have 4 weeks post-cycle. So we have testosterone, total and free. We have luteinizing hormone, follicle stimulating hormone, and then some liver enzymes. So in response to SARM use, we see a substantial decrease, about a 70% decrease in total testosterone and about a 40% decrease in free testosterone. Luteinizing hormone, we weren't able to get data from pre-cycle, but on cycle, we can see that luteinizing hormone, they are within the clinical reference range, and follicle stimulating hormone, they actually fell below the clinical reference range. So this kind of lends to, I'll just mention that this kind of lends to the whole idea of the oral contraceptive use, but it's important to note that even 4 weeks post-cycle, this individual was still suppressed. So that oral contraceptive action of the SARM is continuing even post-usage, which isn't necessarily a good thing, right? And then when we're looking at liver enzymes, they're above the clinical reference range here on cycle, indicating there's some degree of liver toxicity from these compounds. Again, but this is orally administered, so it has to go through first pass of the liver in order to be metabolized. And post-cycle, they do come back down to normative values. So, and then the last thing we'll talk about here is what's happening in the muscle. So this is, again, cross-sectionally, this is what happened. In the black is what is happening with that individual on a cycle. And in the red is what's happening in previously published normative values. So we have a decrease in intramuscular androgen receptor content, but an increase in intramuscular androgen concentrations within the muscle, which is very interesting. So this makes sense. Androgen receptors would be down. If we're saturating the muscle, the body is saying, well, I already got a bunch of androgen-like compounds, i.e. SARMs, activating this receptor. We can downregulate this to try to get it back to normal activation levels, if you will. But this data right here in terms of intramuscular androgen concentrations being elevated above what normal people would be, that's definitely an area for further exploration here, because that's a very unique and never documented finding that we have. But I want to caveat all this, but this is an N of 1. This is a case study. So this is just one individual. So we can't have statistical significance with any of these measures, that we're just reporting what we found. So that data kind of speaks for itself, is what kind of this summary slide says. So I won't reiterate what I just said. And we'll stop there and I'll take any questions.
Nice. Thanks, Tom. Give him a round of applause. Well, I'm sorry that I kind of had to rush you in that last little bit, because that last bit's actually the most exciting part, right?
I know, and like I wanted to set it up so well for everyone to really get into that data, but yeah, hopefully that they hopefully you guys kind of saw some of the cool data we found.
This entire thing's being recorded, so you know, the entire thing's also up for it's fair game for an exam, so they're gonna have to rewatch it anyway. So they should be able to get through that and become SARM experts by the end of it. But yeah, if we have any questions, I think we have time for, yeah, maybe 3 or 4 questions. Yes, Andrew.
So,
Can you hear him, by the way?
Faintly. I'll try my best to hear.
If you don't mind actually coming up and asking, that'd be fantastic. Not put you on the spot.
so are orally active SARM 17 alpha alkylated like AAS?
I'm sorry. Can you repeat that?
Yeah. So orally active SARMs.
Yeah.
Do they have to go through 17 alpha alkylation in order to be orally active, or do they go through something else?
Right. So because these SARMs aren't necessarily steroidal in nature, we still are divulging, trying to figure out exactly how they are metabolized and broken down in tissue. So they're metabolized in the first pass of the liver. To be honest with you, I'd have to look into that more. We're focused more on kind of not necessarily just the metabolism of it, but the end goal of how these compounds are influenced in the tissues that they're targeting. But now that we have some of this data on liver toxicity and the fact that there are some negative impacts on metabolism, that's definitely an area of research that we need to focus more on. So to answer your question, I don't believe so, but that's something that I would have to look further into in terms of focusing on the metabolism, because I'm so focused more on the physiological genomic effects of these compounds in their target tissues. And Stephen might have some more insight into that too.
Yeah. So the cell 17 alpha alkylation, that's the primary modification to the testosterone-based molecules in order to make them orally available, correct? And that also is the primary mediator behind their hepatotoxicity. Yeah, so I mean, in my opinion, like Tom said, I'm not entirely certain about that, but because they're not steroidal based in nature for the most part, I would assume that you wouldn't need to make those modifications to the structure in order for them to become orally available. But yeah, I'm not super familiar with the skeletal structures of them enough, and the ways that you modify or create those structures to make them more orally available. But yeah, it's a good question.
That's a great question.
Cool. Anyone else? Y'all have to come up with questions. You're not leaving here until 3 more people ask a question. Yeah. Come up here and say.
And again, questions both in the clinical sense and the athletic and fitness side are great, because we can talk about all that.
Okay, so if someone say someone took the SARMs for an extended a long time, after say they stop, does it have a certain what's the word, did they lack something, or is it just part of their DNA makeup at that point? You know what I'm saying?
Yeah, or the effects do you rebound?
Yeah, is there a withdrawal stance, a withdrawal period of the not taking it, or
Yeah.
is it click back to regular DNA makeup? Or,
Right.
Yeah.
Did you get that, Tom?
Yeah. So great question. So I'll start by saying that these compounds have different half-lives. So the amount of time that they're biologically active in the body is going to differ depending on the compound. But our data actually lends to the fact that there could be lasting effects, although no research has really investigated those lasting effects. But it's thought that most of these compounds are reversible as far as the negative impacts that these compounds have on the hypothalamus, pituitary gonadal axis, and the effects in the tissues themselves. They are reversible after they stop using. However, our data shows that follicle stimulating hormone, which is responsible for spermatogenesis in men, is actually still below clinical reference range, even 4 weeks after the cessation of use. So there could definitely be some lasting damage if not treated properly. So this also lends to the idea that we need to have proper post-cycle therapy implemented in these individuals, so that they can restart and kickstart this axis, so that they're not impotent or have lasting effects on some of these tissues. But things like those liver enzymes return back to baseline. As far as the hematological measures that we took, there was no real impact on those. Kidney, no impact on those. So mostly reversible, but we need to learn more about some of the lasting effects, and there could be some more and more research is done. And again, this is a hard area to do that research in when it's so heavily regulated.
Cool. Yeah. You know, I think we are actually done on time. So those were good questions, and appreciate it. We appreciate your time, Tom.
Yeah, no problem, guys. And I got my contact information here if any of you guys want to reach out, whether you're going the research route or the fitness coaching industry side, or if you're interested in medicine. Again, I'm working in a medical school right now, so kind of have that unique perspective. So I'm happy to reach out and talk to students and help you guys however I can.
Awesome. Well, thanks, Tom. Appreciate your time. Everyone, give him a round of applause. One more. All right, I'm gonna sign off. It's gonna be recorded. Do you want me to keep this off of like public sectors and stuff because it's unpublished?
No, you're good. It's published in at a conference.
Okay, yeah.
So
It is published in ISSN, that's right. Okay.
Yep.
All right, Tom. Have a good one.
all
I'll
right,
talk
thanks guys. All right, sounds good. Thanks. See ya.